So I'm 1.Alena, I'm from Leiden University, and my interest in chytrids or zoosporic parasites has been basically in population control, food web context. So I've been looking a lot at type of biotic and abiotic drivers that actually make infections more prevalent or less prevalent. So I'm going to try to show you, well, I will show you a couple of examples of that. But first, I want to show you a little bit of a conceptualization. Then I'll do it this way. But yeah, so first, maybe I can actually move that somewhere else that's not on the title. Maybe here is better. Okay. So that's basically what I want to give to you today, because I guess not everybody knows all about zoosporic parasites. Tiny introduction. So what you see here is, for example, a sporangium with zoospores in it and an empty sporangium, which colors really nicely with chalcofluor white and under fluorescence. I will give you a bit of an introduction to SIR models. Is that who's familiar? Well, you can actually correct me, I guess. Cool. So it's not super familiar to everybody, which is good. And then I give you a couple of examples how that actually looks in field and lab observations. Zoosporic parasites. So what are they actually? It's a very broad collection of taxonomic groups, actually. What combines them is that they are fungal and fungal-like. In this case, we're interested in the parasites, and they have a couple of similar morphological traits. What you see here on the screen is an endobiotic and the epibiontic sporangium of the parasite on a host. Both of them are microalgae. And the latest paper that Silk and Albert and Slavek actually just got accepted on the occurrence and diversity of this group. We actually said we include these 13 taxonomic groups. I'm not going to read them out. It's highly polyphyletic. That's a message I want to give to you. They do these epi or endobiontic infections, and they're all, and that's really the combining characteristic, they all have a mobile transmission life stage. That's this tiny zoospore. They are about 1-2 microns with an oil globule or lipid globule, and one or several flagella. And they actually actively move through water. They need a watery phase that they can move through to find a new host. And I think Silke you will show a bit, maybe a bit more. Find it. You have it? No. So of course the life cycles are different for the different taxonomic groups or species, but I'm just showing now here the chytrid example, where you have a combination or two possibilities of an asexual life cycle and a sexual life cycle. The asexual one is that the zoospore, now in the front for people online, the zoospores actually actively swim, find a host through chemotaxis most likely, attached to the host, and then there is also some chemical crosstalk involved there, because sometimes these infections are not successful, they're even aborted. But if the infection is successful, the zoospore will lose the flagella one way or the other, and some sort of a hyphen type system will be extended into the host. The entire host content is digested, taken up into the developing sporangium. Through mitosis, you get, you will talk much more in detail on that, I guess. Through mitosis, you will get zoospores developed, and when it's ready, the sporangium bursts open, the zoospores are released. And then you also have a sexual cycle where you have two compatible attached sporangia. which form a zygote, and then a resting spore. This resting spore is also interesting because it can stay for a kind of an undefined amount of time, so at least several months, potentially even more, before it germinates again and then goes through zoospore formation, and then you get the zoospores out again. And just to give you a first glimpse into the diversity of these are all chytrids actually. They really infect a wide range of microalgae and others. But that also puts me to the next question of why do we actually care about these zoosporic parasites? First of all, they occur everywhere, in every aquatic habitat, even in snow, which I find very cool. And they're globally distributed, and they have this really wide range of host species. Host specificity is a different question, but they can infect seaweeds, algae, cyanobacteria, and also some of the famous biomass production species like Haematococcus in this example. When they do very well, they can produce these intense epidemics. In the Acinella host, they can actually even reach 90%. That's not true of all chytrids. You get everything from tiny little sporadic infections to these huge epidemics that also really make the host population crash. And we'll talk a little bit about that, why that might be happening later. And well, today we're talking about parasites, but chytrids actually, and in most of these zoosporic taxa, they can have a wide range of lifestyles, from truly saprotrophic to facultative parasitic to parasitic, even to hyperparasitic. So chytrids that infect other chytrids sitting on another host. So there's a lot of things to play with. And there's a lot of buttons you could tweak to try and understand when infections are particularly successful. But maybe first, I thought it's good to disentangle a little bit on why parasites are different from predators and what we want to be looking out for. Any ideas? What makes a parasite different from predators? Too early to talk. I'll get you talking later.
No. I can say something, of course.
Yeah, you're welcome.
Well, predator is a one-time kill, and a parasite actually has a longer period of engagement with the host.
Yeah.
Where it doesn't necessarily immediately kill it, but drains energy.
Yeah. You got my first sentence. Perfect. So indeed, the parasite needs the host to survive until it's finished with its life cycle on the host. Otherwise, it's also game over. Whereas the predator actually needs that the prey dies at some point before it ends in the stomach. Also, so that's the interaction duration that you just mentioned, but also the number of prey or host affected. So one parasite individual will be linked to one host individual. There is one lion will eat many zebras in its life. There is also difference in terms of morbidity versus mortality. With a parasite, you don't necessarily always have to die. Yeah.
And online, there's a question: are parasites maybe opportunistic?
Opportunistic
versus non? I don't know. Can you say something about that?
That's a good one.
That's a question.
Yeah, opportunistic in. I'm actually not sure. I mean, as a predator, you can also be an opportunist. I would have to table that one too and think a bit more about that. Thanks for the question, though. Both can regulate host population dynamics and size, but parasites can make this massive epidemics and really drive a population to extinction, at least locally. For predators, you would enter these predator-prey cycles much more easily. So there is, yeah, a timing and effect size difference. Yeah, parasites rely on transmission and encounter. Predators mainly on encounter and ability to kill. That one.
Maybe it's on your list. You're going down the list. I was thinking about, I'm not sure for kitchens, but if you think about pathogens like viruses, they can also give benefit to the host.
Yeah.
So they
no,
can
I
add
didn't.
traits actually, like some defense against other pathogens, or maybe increased photosynthesis.
Yeah, no, I did not include that. So that's a very nice point to also look at the beneficial side of some parasites, at least. So this difference in whether it's transmission or encounter determined also has influence on the adaptations of the host that are then useful in the defense, and also on the evolutionary trajectories and potential for co-evolution. And then one other thing I want to point out is that the trophic position of parasites is always very tricky to assess with stable isotopes. So you really need to get a good view on life cycles and host range to position parasites securely in a food web. Okay, now let's get a bit more to the drivers. What do you think? What do you see here? Well, something with disease. And these are years, and you see cases.
Flu in Iceland?
Flu is correct. bit closer to home. I think this is a very good giveaway of what it is not. Yeah, indeed. The flu in the Netherlands. What strikes you if you see this? Yeah, so you have these peaks, but then also it stops at some point. So there's the transmission in between, or not so much. Go ahead.
No, it was just
maybe you also see that the peaks are not all the same height. Sometimes they don't occur. So indeed, very seasonal. It also stops. And transmission doesn't really seem to be equally successful everywhere. Why would that be? Why don't we just get the same nice, like we have people who always get the flu, people who never get it? Or why do we get it again?
different strains.
Different strains, indeed. Climate conditions, indeed. So the other things that the environment around the host and the pathogen, in this case, parasite. I'm going to be very loose with the terms pathogen and parasite, so apologies for that. Indeed. So there's other factors at hand that modulate this interaction.
Well, you should explain 2021.
Yeah.
Anybody?
Who wants to say something about why was there no flu?
Why was there no flu in 2021? Masks. Masks, one and the other?
Yeah.
We were all at home.
Yeah. Social distancing works.
Well, nobody was exposed.
No
Yes, records also, yeah, because if everybody was at home, maybe also personally in charge of the recording.
I think they actually still would have picked it up, because now it's yeah, you have quite a hospitalization rate also for flu. It indeed. I think in some countries, then the 2022 peak was actually especially strong because there was whatever basic immunity you would have built up over the years. That was not so helpful anymore in the next year.
Is the zero also because you would maybe tag flu as COVID?
Yeah, I think given with all the testing in that year, I would not, well, at least you would not miss these peaks. You should pick up something at
No,
least.
0 is quite, I mean.
0 is really strange.
Yeah, I'm wondering.
Yeah.
But 2020 was also lockdown, huh?
That's the winter season.
No, but the lockdown started in March.
Yeah.
Yeah, okay, so the peak is not, and it's stopping the peak, it's not this typical modern curve, but still, I mean, 0 from 0.
Yeah, I also don't know whether it's a true 0, huh, given the yeah.
Also the number, I think it's maybe look at it before. The number of specimens, it cannot be that totally future.
No, that is there is a factor behind
Yeah.
it. So, yeah. All right. So one way to look at the influence of the environmental factors surrounding the happening of the disease is to actually look at a compartmental model of disease spread, which is this SIR model. And that's not a new thing. It's actually from 1927 already. And what makes it particularly useful is that it's deterministic. So you can really use it to run simulations or to calculate rates, to get an understanding of what processes are going on, and maybe even a mechanistic understanding. It's very versatile, so you can adapt this model to your specific disease in most cases. And you can add relatively simple equations to it that actually allow assessing environmental effects, not only stationary ones, but also dynamic ones, like seasonal changes in infectivity, for example. I'm just going to walk you through the model, build it up with you. So during a disease outbreak, you have a total population N, so that would be all of us in this room. And we can actually put them into 3 compartments. So S would be the susceptible ones, that's the naive population, which can be infected. So they're not immune. Once you get infected, you move to the next compartment of the infected, and then hopefully you also recover. So you move to the recovery compartment. And I already said, you have to move from one to the other. So from S to I, you have disease transmission, and then from I to R, you have recovery. You might lose immunity, so then that would also mean that you can move back to the susceptible. That's partially what you see when flu strains evolve differently than we evolve, so we become susceptible again to that disease. And what this model does really nicely, it allows us to calculate the rate of change in each compartment over time. And in the end, we get numbers of individuals per time step. So not only can you say like how many are in which compartment at some point in time, but also like how fast do people or individuals or hosts move through these compartments. And so that means that you have these differential equations of the compartment changing over time, so the rate of change, which in this case, for example, is proportional. So the susceptibles to infecteds, and then the proportional transmission rate in between. Here in the infected, you of course gain whoever gets infected, but you lose whoever is recovering. And then in the recovered, you of course gain everybody who is recovering. So it's not all that complicated. Hopefully.
Not that, no.
Not that?
No, it's just that there are the recovered, but also the death.
So in this one, at the moment, nobody has died yet.
Okay.
We'll get to that. It's a very benign disease. You just get the sniffles and then you recover again. And often we are, of course, very much interested in this transmission rate. And ideally, you would have 2 informations there, namely the contacts over time or per time point, and then also the probability of getting infected, which is not, I mean, sometimes you might get into contact with a virus, but your immune system is good enough, you can fight it off, you don't get infected, right? So it's not always a 1. It's luckily, it's most of the time, it's much below that. And then recovery is actually linked to how long you are infected. So how long is the infectious period? If you have complete recovery, so if there's no deaths included. Yes. And then, of course, we get to this basic reproduction ratio, which I think everybody still remembers from Corona times, and then the daily news, trying to figure out whether it's now bad or not so bad. But as a refresher, the basic idea here is that to put a number to how likely it is or how many secondary infections you get from an average first infection. So if I'm sick, how likely is it that you will actually get it? Under the assumption that the population is susceptible, so not vaccinated. And well, you can do some gymnastics, but in the end, the idea is that you can actually calculate it from the transmission rate and the recovery rate. And it gives you a number around 1. So if it's 1, then it would mean that the infected person get 1 new infection. If it's above, then you can move towards an outbreak, depending on how high that number is. And if it's below 1, the disease is actually slowly on the way out of the population, maybe even to extinction. And then you can actually put it in a model. You can model very nicely that if you start with a susceptible population here in red, you add a disease, an infection to it, it spreads through the population. So in green, you see the increase in infected. At the same time, your susceptible population is going down, which also means that the disease is less, also the infected component is going down, and the number of recovered in this case is going up. So now you think that's very nice, but what does it have to do with drivers? What is she talking about? It actually becomes interesting when we can put our environmental drivers onto these rates and manipulate them. Of course, this all does not happen in a vacuum. So like Laura already said, there's births, there's deaths. There might be immigration, there might be emigration. So these are all things you can quite easily add on to the model, right? By adding to the differential equation, birth rates, mortality rates, which might be the same for each compartment, which might also be different for each compartment. But also the entire population might change, and then you would need to figure out how you allocate this inflow to these different compartments. So if you have immigration, which is completely unvaccinated and susceptible, they will all end up here. But if you would have vaccinated immigration, then they might actually all more move to this pool, so they'll not add to the susceptible ones. And of course, there's also potential for emigration. But it really gets interesting when we can start playing with these 4 rates and add whatever we are looking at to that: temperature sensitivity, salinity sensitivity, seasonal variation, for example, in light, if you're interested in a plant pathogen. That seasonal variation in light would affect your host, and then through the host also affect the parasite. So that's actually where this model also becomes interesting for the question today. And then that links very quickly to the disease triangle of these 2 seminal papers in the 1960s, where they already made the connection that, yeah, you have the host-parasite interaction, of course, but they are also both exposed to a shared environment. This environment has a direct effect on the host and the parasite, but it also has an effect on the interaction strength of those two. Yeah, let's just try whether we can do that. This is a Whoop lab. It's like Kahoot. If you take a picture of the barcode, not barcode, QR code. Thank you. The only thing now is that I have to get to the other screen. I don't want to stop sharing, but I want to get out of the full screen mode. I am. No, go away. Yes, we can work with that. Most likely we can work with that. Let's see. Okay. It looks like everybody's in already. Otherwise, you can always go through the code. Just go to Vocab.
Oh, and it says, to me, it says in Dutch, deel uw ideeën, and that means share your ideas. I don't know if that's right.
Yeah, but you can also.
Okay, that's good. Why do I have it in Dutch?
Because you have a Dutch one, probably. That's the language of your phone.
It's Italian.
I have it. You have it in Polish?
Yes. I got it. All right, but yeah.
No.
I didn't consider that I, of course, have to scroll down. Cool. I think we have a lot of ideas, which is good. You're not ready?
Just get started. No.
Ah.
If there is something that we really miss there, it's somebody that want to add something that is not there yet.
I think we have a very nice collection.
We have a nice one.
So indeed, basically why I wanted to get you onto that is that you quite quickly find out there is a lot on both categories. And then sometimes there's also things where you might even start doubting a little bit on whether it's the one or the other, which is not necessarily bad, but it forces you to think on where in the model could you add that on as an extra factor or as a modulator of one of the rates. We'll cover a few of them, mostly on the abiotic side, but it also highlights that there is actually, in my opinion, a big knowledge gap. For example, I saw here the host microbiota is there's not all that much for phytoplankton and chytrid interactions, for example. There we're going there. The effect of competition, yeah, that's also an interesting one, right? Where do you add that one? Is it competition on the host level or on the parasite level? Predation, same. Or maybe hosts that are in fact easier to predate on or more nutritious and nicer to predate on. Yeah, so thank you. I'm gonna try to get back to the back to the other screen. How am I actually doing for time? Oh no. Cool. if anybody actually wants to play a little bit with that, there is this really nice document of the World Bank, which also gives you an introduction with an Excel example. So you could actually work through it and then run these models in Excel yourself. And obviously you want to do that. And different tools for real, but it's a nice introduction just to see how these things play. But yeah, let's look at a few of the drivers where I have data for, and there's definitely more out there, but it's just a little bit of a selection. If you look at the transmission in disease ecology, you get a distinction between density-dependent and frequency-dependent transmission. Who's aware? Who knows that? Just pointing you out. The density dependent is quite intuitive. The denser the population, the more easy it's for the parasite to find a new host, and you get faster transmission or more transmission. That's true for all the hand-shake diseases, pretty much. Like if you go into a club, it's crowded, somebody is having a cough, it's quite easy to catch it. Frequency dependent actually is independent of the density of the host population, but it depends on the proportion of infected individuals. So it's more often the case in vector transmitted diseases. So if you have mosquito carrying malaria, it doesn't really matter whether the frequency of the transmission rate doesn't really matter whether there's 100,000 people in front of that mosquito or 10. And it's also the same for like sexually transmitted diseases. For the chytrid and Acinella, we get this picture. So here you have parasite abundance on the y-axis and host abundance on the x-axis. Which one do you think would it be? Density? Or yeah, you're excluded. Density or frequency? Does it cap off? Does it matter for the parasite how many hosts there are? It does. So it puts you up in this one, right? Cool. So that would already be one first modulation that you can put on the transmission parameter, for example. Then another example. This is from a lab experiment with Acinella. So that's a diatom host and the chytrid associated to it across 5 temperatures from 1 to 21. Here you have the net production of infected cells. And that's the production of infected cells over a time period of a week, if I remember correctly. And what you basically see here is that at 1 and 21 degrees, there is hardly any new production of infected cells. But then in the temperatures in between, you get this nice tolerance range curve. The usual thermal reaction norm. With, in this case, the interesting distinction that at 1 degree, the disease all kind of disappears into resting spores. Whereas at 21, it doesn't do that. It just died. Actually, it couldn't deal with the heat. So where would you use that information in the SIR model? And maybe how? Multiple options, right? So the easiest might be to have a temperature-dependent transmission rate. Also here, having these different life stages where you have either death or you have this resting spore is also something you could play with. So you still have the parasite in the system, but it's not actually doing anything. So how do you translate that into a transmission? How do you translate it into the pool of the infected? Is there any recovery in that case? Maybe. So that's ways to play with it. But also ways to think about it that there might be refuges from disease, right? That's an example by McKindles and co-authors looking at conductivity and infectivity of the percent increase in infected filaments per day in the ParAqua-COST system across a range of conductivity, so not directly salinity, but just electrical conductivity of the water. Where they see if the conductivity is quite low, you get really nice spread of the disease, a lot of new infected filaments per time. And if you increase the conductivity, that dramatically decreases and goes even to, I think, below 0 here in this case. So you're actually losing the disease. That would be R naught under 1. So also here you could modulate, right, the transmission with the transmission factor or parameter based on that knowledge. From the same system, same paper, actually looking at water movement. If you think about zoospores, why would water movement be interesting in that case? We have these tiny zoospores swimming around, trying to find a new host in this big ocean.
Directional transition. They can't swim against the current.
Yeah, they just kind of get carried somewhere else. Indeed. So again, here on the x-axis, you have the water, what is it now, stability? No, yeah, stratification, I think. And then the increase in infected filaments per day on the y-axis. And you see they can deal with some water movement, but then at some point it just stops. If the water moves too much. The zoospores, for some reason, can't attach. Are you going to show that? Ish?
A little bit.
Then I'll park it to yours. Huh? You can also think about not physical things, but pollutants. This is an experiment by Nandini Raman working with this really nice subastromal system, Silke. Very beautiful, very well behaved also, so I can really recommend it. And what we did is we exposed host and parasite co-cultures to a range of diclofenac. And why diclofenac? Diclofenac is globally used, and in many countries you also observe in the surface waters concentrations that are above guidelines. So we were interested in if it's out there, we don't know what it's doing. It has something to do with some receptors that are also occurring in chytrids. So we thought we might be seeing an effect. And we also did see an effect, but then only at really high diclofenac concentrations, like what you would get in the global south, maybe in the direct outflow of a hospital. So it's not very natural situations where we found an effect. But what was still interesting is that the host actually was a little bit better at dealing with that amount of diclofenac than the parasite. So there was again a situation where it was really not ideal for the host to survive long term as a healthy population, but the parasite was even less able to deal with it. So you can get these pockets of windows of opportunity for disease-free growth. Need to show this?
Yes.
Yeah. So that would be my only biotic example, but then I'm also going to park it for.
No, I'm not going to show.
You're not going to show it?
No.
Okay. So Thijs did this really nice experiment using a Synedra culture. So Synedra are diatoms which are kind of a long needle. They're not edible for tiny zooplankton rotifers. But if you infect a Synedra with chytrids, the chytrids make zoospores. These zoospores are then actually edible for the rotifers. So right, so that's the microloop that a couple of you already mentioned today. So upgrading or rerouting of energy and nutrients that would not be available for a certain consumer class actually kind of gets rerouted and becomes available, and maybe even upgraded. I think Serena will be talking about the upgrading. Perfect. And what Thijs did, do you want to say it yourself?
We did.
Well, I can't remember. No. So we exposed infected. Did we use Plantotrix? Yeah, that's the wrong graph. So Plantotrix, but same, long filaments. We exposed infected Plantotrix to different densities of consumers, and were a little bit disappointed that in terms of prevalence of infection, we didn't really see that much of a difference at all. The disease was just happily infecting everything. So this is 75% here, so it's way above 75% at the end of the experiment. But looking at the zoospores, we saw this quite strong difference. So this is without any consumers, and then with consumers, you see this reduction in zoospores. It didn't mean anything for the host because there were still way too many zoospores to make any dent in the prevalence. But again, here, that's ways to think on how can you modulate that? How would you enter that into an SIR model? When does it actually matter? And the outcome along these lines. And all of that kind of 4 years ago or so brought us to the idea that we got interested in the tolerance ranges. Right? So you saw that chytrids and hosts don't necessarily react the same to the environment. And the tolerance range is a good way to visualize that. So here on the x-axis, you can think about any type of stressor that you want to think of. I mean, temperature is always an easy one. And then the host has a tolerance range to that, the parasite has a tolerance range to that. If they are both completely equal, you would assume that the parasite can infect in any case, right? But they might not be equal. There's a thermal mismatch hypothesis out there, which basically says parasites are in general smaller than the hosts. They have faster generation times. They have higher evolvability or adaptation capacity. So they should have a broader tolerance range than the host. So you would actually get this shift. And of course, they make it a little bit more complicated, but in a nutshell, it comes down to that. What we observed in the chytrids often is actually the opposite, that the chytrid has a narrower tolerance range than the host, which gets these interesting refuges of environmental conditions where the host can survive, but the parasite is dead or inactive. But then we live in a changing world, right? Climate change, global change, and general evolution. So that means we're not necessarily keeping these refuges where they are now. And then the question is, if we change the range of, for example, temperature that something is exposed to, will we then lose some of these refuges, but maybe also gain bigger refuges at the other end? So that is, of course, also what makes diseases often quite unpredictable. If you remember a couple of years ago, the general view was with warming, the world would also get sicker, but that's not entirely one-to-one true, which has all to do with losses and gains of potential refuges. But in reality, of course, we have multiple drivers acting at the same time, changing in different ways. So I'm just showing you one graph from my PhD work in Lake Marseille, again on my favorite Astionella and the chytrid. This is 1.5 years of winter bloom, summer bloom, winter bloom, summer bloom. Well, early with the parasite abundances. So the yellower it is, the more there is, and then infection prevalence. And at the same time, we also measured a range of nutrients, temperature. We calculated water stability. We measured light intensity and light extinction. And then modeled host and parasite abundances and how they are modulated by biotic or abiotic factors at different depths. And basically what comes out of that is that both biotic and abiotic play different roles. Not always the same set of abiotics was present or driving anything at different depths, but also the intensity or the strength of that interaction changed with it. And of course, there's many layers to it because there's a production layer for the Astionella, and then the Astionella sink out, so it has inherently to do with the water column stability. So you get all these different trajectories of what is influencing disease and disease spread. So it's good to keep in mind that refuges are not universal, they're very interdependent. And trying to tackle that through simplifying models might be one way forward. Getting good data is definitely another way forward. It should be combined. And that actually brings me to my, if we have a little bit of time for discussion still. Yeah, we do, 5 minutes. To maybe 2 different questions that we could look at. So one would be like if environment, or well, because environment is inherently multivariate, how does this affect disease refuges? But maybe for this round, it's actually more interesting to look at the second one. So how can we translate this type of ecological knowledge into applicable solutions, so that, or applications, right? That would be interesting for you. And what are we missing, both in conceptual understanding, but also in terms of the type of data that we gather, or compartments that we are completely missing. And with that, I'm going to shut up for a moment. If online there's questions, I'm also happy to hear.
Yeah, and they can also ask directly or put it in the chat, and then I ask it for them.
True.
Then there is. So can I kick off, Alena?
Sure.
So about environmental drivers, you mentioned already, like you have the effect of host and parasite, right?
Yeah.
How would you, because you also showed some field data, so how would you do lab stuff? So what are ways to disentangle the effects on parasites directly versus indirectly through the, because you have some effects on through the host responses and some are directly on the parasite. How can you differentiate those?
And in many cases, it is difficult, of course, but going through specific traits is one way, I think, to really measure trait outcomes, like response of, for example, for the Astionella chytrid system, there's of course direct effects of temperature both on the host, so host only, you can measure that, but then in the host parasite, you can also measure swimming velocity of zoospores across temperatures, and then you would have a parasite direct measure. You can do very controlled infection experiments where you really know how many zoospores you add and how many hosts you add across temperatures. You would get a better idea of the infection probability. So that you can narrow down, but then that means, of course, that you have to do a lot of different experiments to kind of get the complete picture. That's also why there's often still a bit of a black box approach we see this increase over time, which is a combination of direct and indirect effects. That's much easier to measure. So if you really go down to specific traits or specific processes, you can do that. Not all of them in all the different systems, but you can do that approximation. Yes, Elena.
Yeah, about this point, I was wondering, I mean, besides running different experiments, well, you always have a control, I guess, and often without parasite. But what about statistical tools? I mean, you can run multivariate statistic with and without parasite, or you can use like GAM models, disentangle a bit the different factor and use the host abundance separated from the parasite abundance. So I would like to hear a bit also maybe about this kind of tools that ecologists love to use about this kind of analysis.
Yeah. I'm too much of an empiricist to completely let go of having some sort of a view on it. I think you can get very far with a path analysis or with multivariate statistics. But you will always there will always be a reminder of that you cannot really allocate. I mean, you have the host on your control and then the infection, the host parasite co-culture. And so for the ones of you who are not familiar with chytrids, there's no way of having chytrids, these parasitic chytrids without the host. So you always have to have them in the parasite host co-culture. Where it becomes tricky to really disentangle which part of the variation is now down to direct effects on the parasite, on the maybe zoospore stage, but also on the sporangia development stage. And what is mediated through the host, because the host is having, I don't know, producing heat shock proteins, and that primes it to be better defended against infections. Not very satisfying answer, I'm sorry.
No, well, but I mean, there is always a certain amount of the variable that you cannot explain, and I fully agree. It was just get a bit different, maybe, as I say, tools that can be used a bit also. I mean, after the experimental design, because often, I mean, the experiment then is done, we have a data set and we need to analyze it, and we have to deal a bit also with choice that have been made, I mean, maybe months earlier.
Yeah, it is. Maybe I would see more opportunities in going then away from the statistical modeling and then go to proper, how do you call that? Theoretical modeling, where you could test different processes. So you could actually take the SER model and then say, like, I'm going to assume temperature does this to my zoospores, so that has an effect on the transmission rate, and I'm assuming temperature does this to my to my host population. And then you can actually simulate that. So by assuming certain processes, you could see whether that actually gives you the same patterns that you see in your experiment, and then you could allocate it more cleanly to different processes. Not sure whether you as theoretical ecologists want to add to that, because I'm sprouting some stuff here.
So how would you deal with that? So in your case, or maybe also with your models, right? How do you deal with this? You have a model and then you have these parasite host parameters and parasite parameters, and you want to parameterize it, and the only thing you have is literature, right? Experiments from others, fieldwork, etc. So how do you get your rates out of that, and maybe sensitivities to certain environmental drivers?
I think that people often expect too much from the theoretical models.
Damn it. I was just trying to push it off.
It's like, you know, it's actually, it has to do with being familiar with it or not. It looks like, oh, very nice, complicated, this will be the solution. But if you think about so if you look at this SIR model, so one thing is like, where I already see a mismatch is that you in the SIR model, when an individual gets infected, it immediately contributes to new infections. And you describe very clearly, like, okay, we have these zoospores, they attach. There's a whole, there's a whole development, there's a whole process underlying that. So we have one parameter to actually parameterize that contribution of an infected individual to the epidemic.
Yeah,
and so how do
it's
you
not online.
choose that?
Yeah, so one way we dealt with that with 1.Alena was actually to model the production of zoospores in the SIR setup separately. So it's an outflow from the it's a function from the infected, which makes a compartment zoospores, and then from that compartment you have again it actually went back to beta, so to the transmission. And in that way, so by adding compartments that are important for the biology, you can tackle it. It's not perfect. The time delay is still an issue.
And I think that so what you can expect from that model, from that type of models, is more like qualitative insight and maybe semi-quantitative.
Yeah.
But never the type of quantitative predictions that you actually would want to have to test in experimental setups. For that, you have to be much more specific about like what's the process that you're actually considering.
But maybe then I'm a little bit too naive, but I was actually kind of thinking that the SIR model can give you a tool to look at potential processes and then kind of trying to figure out which ones might be more interesting or more important to look at empirically then to actually get good numbers.
No, that's absolutely true. Yeah. And it depends also on like what's your focus on the model?
Yeah.
Is it the occurrence of an epidemic or the start of an epidemic? Is it your R0?
Yeah.
The whole theory about R0 is very well worked out and it's very general. But that part is really
developed.
It's more robust than, for example, like prevalence.
Yeah.
Prevalence depends on much more than just whether or not an epidemic could start.
Yeah. We might have to switch soon.
One more question.
Okay.
One more question. There was question here as well. Mine's a very basic one, so quite quick. But I saw that when the chytrids are infecting filamentous algae, they're sort of targeting one cell within that sort of string. Is there any way that they can infect nearby cells through the hyphae and stuff like that, or is it just literally that one section will die and then the spores go out and find new ones, or is there an internal way of it?
It's just left the room. Shall I take it on you? No, I mean, it actually depends. So with Plantotrix, you see that the hyphae work through several cells. For example, in all of the xylose chains, there's enough separation between the cells that the chytrid still only attaches to one cell.
Depends on the species.
Yeah, and then also Plantotrix seems to be vulnerable at the growing ends and not at the sides. So there seems to be enough defense that the chytrid cannot digest its way through the sheath layer. That makes it so special. So also the reason I think why we saw this high prevalence despite the drop in the zoospores, because it was just not like, I mean, the opportunities to infect were much reduced, so to speak.